Good evening, everyone. I, Khushbu, welcome you all to our webinar today on how millets talk to fungi, a transcriptomic perspective. And our speaker for today is Vaishali Chakrabarti. She is a Senior Manager Solutions at Strand Life Sciences. I welcome you, Vaishali, and she has an experience as an intern at the ICPO, ICMR Noida, as an application science specialist at Strand Life Sciences, Bangalore. She has worked under multiple avenues as a product development, product manager, data analysis, and curation. And now she's the senior manager of solutions at Strand Life Sciences. And her educational background includes her bachelor's degree in biotechnology and a master's degree in bioinformatics. And I welcome her team members. Uh, Dr. Srikanti, she's the Associate Director, NGS Applications, and she leads the NGS Applications Division at Strand. Anoksha Bhattacharjee, she's an application scientist working on the development of Strand NGS platform. And Pooja S. Math, she's an application scientist with expertise in handling commercial research projects for several clients working on millets. Um, I welcome all of you today, and I would like Vaishali to please take the session forward.
Thanks a lot, Khushbu. Let me first share my screen. Khushbu, can you confirm if you were able to see this movement?
Yes, ma'am. Your slides are visible.
Okay,
Yes.
okay. Thanks a lot. So thank you, everyone, and thanks, Khushbu, for starting the session. Thank you, everyone, for joining today's session. So we'll be talking about millet, the symbiotic relationship between millet and fungi. And so I've kept my video on, but I'll be switching it off now so that there's a seamless internet connection. And after the presentation, we'll have a Q&A session where all the panelists you just now Khushbu introduced, they will also be interacting with you in the Q&A session. Sorry, 1 minute. Yeah. Okay. So before we start the analysis discussion, I would like to provide a brief introduction to Strand and our range of services. The Strand Life Sciences is a prominent academic spin-off from the Indian Institute of Science, Bangalore. And we have over 2 decades of experience in the genomics industry and are actively engaged in a variety of business areas, including research, product development, lab services, clinical diagnostics, assay development, clinical research, among others. Strand provides an extensive range of services, including both NGS and non-NGS based solutions. In addition to the diverse DNA and RNA-based NGS services highlighted in this slide, we also offer specialized microbial NGS services, including metagenomics, 16S rRNA, and amplicon sequencing. For our non-NGS offerings, they include GWAS, methylation assays, and agrigenomic consortium arrays, among others. We have a CAP and NABL accredited lab with all the latest tech that you can see in the slide, including the NovaSeq X Plus sequencer from Illumina, as well as a nanopore sequencer that is the nanopore P2 Solo. Now, our bioinformatics services are well established for standard organisms such as human, mice, and Arabidopsis. However, we also have significant experience working with non-model organisms such as microbes like viruses and bacteria, fungi, protist, plants, different varieties of plants actually. And in addition to the most common sample types such as blood and tissue, we also have successfully contributed to numerous projects involving samples extracted from CSF, soil, plant material, FFP, and other diverse sources. Now, since our inception, Strand's bioinformatics unit has embraced the philosophy of de-skilled decision analytics to empower biologists. So most of our software products are built around the principles of access, visualize, and discover, collectively known as EVADIS, which is a patented platform. Now, this approach integrates a range of functions and features into a seamless framework, allowing biologists to dive straight into analysis, visualization, or modeling without worrying about the underlying statistical or mathematical complexities. The Strand Engis is a comprehensive tool that supports a variety of analysis, including DNA, RNA, small RNA, chip, single cell, and methylation sequencing. And also it offers end-to-end solution covering everything from alignment and stats to gene ontology and pathway analysis for biological contextualization of results. These are our strengths, and our tech support team is available around the clock 5 days a week, and they are always ready to assist you if you need any help getting started. So if you haven't had a chance to try out the tool yet, feel free to request a trial license and explore its capabilities. We also have no hidden costs. So once you purchase the license, everything including annotations, software updates, tech support, everything is included within one license. Now let's talk about millets. So these small seeded cereals, part of the Poaceae family, are not only nutritious but also incredibly resilient. They have been extensively cultivated in arid and tropical regions, and they are actually one of the first crops ever to be domesticated. In fact, the historical records show that millet consumption dates back to the time of Indus Valley Civilization, which is around 3000 BC. So what makes millet so important? It's their remarkable ability to thrive in diverse conditions and their resilience to climate change that position them as a key player in the global food security. And recognizing this potential, the United Nations declared 2023 as the International Year of Millets, following India's proposal, which was backed by 72 other countries. Now, if we look at the nutritional value, take foxtail millet, for example. 100 grams of it contains over 10 grams of protein and more than 30 grams of calcium. So that's about 30% of your daily calcium intake in just one serving. So no wonder more and more people are turning to millets as a preferred choice over traditional grains like rice and wheat. And it's not just about the health, it's also about the trends. In the last year and a half, you would have observed that millet-based cafes and eateries have been popping up. And you also would be seeing more millet-based dishes featuring on the on cooking channels in YouTube. So it's not only a sustainable and nutritious choice, but it's also gluten-free. If we look at the millet market, it was valued at $11.3 billion in 2024, and it's projected to grow to $14.43 billion with a steady annual growth of 4.6%. So it is clear that millets are not just a food trend, they are here to stay. Now, as I mentioned earlier, we generated data for millets and other crop varieties and we have generated as well as worked with the data. But for confidentiality purposes, we have chosen to use a public data set from this Springer paper for this demonstration. So I would like to take a moment to acknowledge and thank the authors of this publication for making the dataset publicly available and allowing researchers like us to benefit from their work. Arbuscular mycorrhizal fungi, or AMF, they belong to the phylum Glomeromycota, and they establish a mutualistic relationship with over 80% of terrestrial plant species. Through their hyphal networks, these fungi facilitate the uptake of essential minerals, particularly phosphorus, and then transfer those to the host plant in exchange for carbon sources. Now, this symbiotic relationship not only enhances the nutrient acquisition for the plant, but it also improves the plant's resilience towards various abiotic and biotic stresses. Hence, it plays a very crucial role in the growth and survival of the plant across different ecosystems. Now, this symbiotic relationship is very well studied in model plants like Arabidopsis and Medicago. However, in this study, we aim to understand the mycorrhization in one cultivar and two land races of foxtail millet via a comprehensive transcriptomic analysis. We selected 24 samples from this study. These samples were on an average of 10 GB per 10 GB per sample. So totally approximately 240 GB of data. And the data was downloaded from Geo via the SRA toolkit. Here we took 8 samples each of two land races, IC36 and Hanavalval, and one from the cultivar, TT8. Just wanted to give a brief context. A cultivar is a plant variety that has been selectively bred for specific traits, whereas a landrace is a traditional locally adopted variety that has evolved over time through natural selection. Now, first 12 samples from the 3 varieties, that is the ones shown as green plants in this slide, are inoculated samples, and they were inoculated with the fungi Clarodiglomerulus etunicatum, which is also a species of AMF, and it is the most commonly associated with a variety of plant. I just wanted to take a moment to dive into the annotations, as they are truly the starting point and one of the most crucial parts of any NGS-based data analysis. So at Strand, we provide annotations from over 17 public sources for a wide range of organisms. These include both model and non-model species and cover everything from reference assemblies, genes and transcript annotations, dbSNP, GNOMAD, dbNSFP, to gene ontology IDs, gene sets for gene set enrichment analysis, and pathways from Wiki, Bioseq, and more. So far, we've packaged annotations for 121 organisms, including 39 pathogens and approximately 20 plant species. And because we also work on client-based service projects, these annotations are frequently used in StrandNGS as well as in other analysis pipelines like Dragon. In StrandNGS, you'll find the prepackaged annotations for different organisms available as a one-click download in Annotations Manager, which you can see here on this slide. For instance, the reference for foxtail millet, that is Setaria italica version 2, was obtained from NCBI with the genes and transcript data sourced from RefSeq. The gene ontology data, or GO data, for foxtail millet was also available in NCBI since end of 2023. And we downloaded that, then combined it with our reference annotations, and then added it to the annotation manager. In this case, we were fortunate to have access to the most up-to-date annotations from NCBI, and our dedicated annotation curation team processed and packaged them for StrandNGS. But what if you're working with a species that doesn't have a clear annotation source? Not to worry, we have the expertise to create custom annotations, including de novo assemblies from public sources. And we can generate annotations based on your specific needs. These custom annotations can be used not only in strategies, but also across other pipelines. So whether you're working with a well-documented species or more obscure ones, we've got you covered. This slide gives an overview of the key analysis options that are present in an RNA-seq workflow in Strand. The blue-colored boxes are options with respect to alignment, including QC checks, filtering bad quality samples or reads, and aligning reads. The yellow boxes show options focused on quantification of genes, normalization using different methods, and filtering of genes with low expression values. The gray boxes here are directed more towards statistical tests, clustering, and different dimensional reduction techniques like t-SNE and UMAP. You can also find correlation between samples and genes, as well as build prediction models. Along with these gene expression-based analysis options, there are read-level analysis like SNP, splicing, and gene fusion detection, which are also part of the RNA workflow. So this tool will offer you a guided workflow for those who prefer a structured approach, or if you want a flexible option to and want to customize your analysis needs, that can also be done to suit your needs. The RNA-seq workflow in Strand, it offers flexible data import options, as you can see here. You can begin your analysis by importing raw unaligned data, such as FASTQ files, or you can import already aligned BAM or SAM files. And if you don't have either of these, you can also import quantified raw count or normalized expression data present in spreadsheet format directly into the tool. And no matter where you start with your data import, all the essential analysis options that are needed to interpret the data and draw biological inferences will be available to you. This slide shows an overview of the steps that were followed to replicate this study. The downloaded samples, that is the raw FASTQ files, were imported in Strand, where we looked at the sequencing quality by checking the pre-alignment QC plots present in the tool. The pre-alignment QC plots have plots where you can check and assess the base quality and base composition of the reads. And this further helps you to decide the parameters that you want to set while running alignment. For alignment, there are three options in Strand. For RNA alignment, there are three options in Strand. And we went with the most comprehensive option and ran the alignment against transcriptome and genome with novel splices. Since in this data, there were no adapter sequences or UMI sequences, the alignment was run using the default parameters against the reference genome. And once the alignment was complete, the post-alignment QC plots were checked, followed by running quantification using DE-seq as the normalization method. There are other normalization options available in Strand as well, like TMM, quartile, percentile, etc. DE-seq was selected to keep it similar to what was used in the paper. The differential gene expression we performed using two-way ANOVA, and Benjamin-Hodgberg was used for the multiple testing correction. Finally, biological contextualization was performed by running gene ontology or GO analysis and pathway analysis. Now, in this slide, you'll see a snippet of the multi-sample alignment report, and all the 24 samples that we had taken had a good alignment percentage of approximately 87 to 96%. The alignment report here, being multi-sample, allows us to quickly check and compare the alignment across the samples. Along with this, you're seeing three plots here. These are for taken for one of the samples for depiction purposes, but more or less that trend was same and followed by this same and followed by the all the samples. The alignment score distribution plot on this right corner, it shows that the aligned reads have an alignment score of 90 and above, with approximately 35 million reads having the perfect alignment score of 100. Next, the reported match count plot. This shows that majority of the reads have a match count of 1, which means that majority of the aligned reads are uniquely matched aligned reads. Now, we can see this in the alignment report as well for this sample, where 94.2% of the reads have aligned, and out of that, 91.6 are uniquely matched reads, indicating that majority of the reads do not align to multiple locations of the genome, and the alignment results are of high quality. Now, if we look into the genic region plot, as expected, the majority, that is 53% reads, are aligning to the exonic region. However, interestingly, 32% of reads are falling in the splice regions, and about 1.3% that you see here are in the exon-intron junction. So this can be mostly accounted for if we look at the alignment report again, where for this sample, where close to 1 million reads are translocated, another million both made flipped, and a few 100,000 are split reads. So these plots do indicate that these samples may be good candidates to look further into differentially spliced regions and gene fusion events, although we have not included them in the scope of this study currently, but it's something that we will be extending further in the future. Principal component analysis, or PCA, was performed on the samples to, this is post quantification, to check the quality of the samples. We saw a clear separation between the inoculated and mock samples, and good clustering can be seen within the samples except for Hanavalval. However, since they were still within the planes and were not exactly outliers, they were retained further for downstream analysis. However, here at this point, if you find any outliers, you can always select and remove that sample for your downstream analysis. Now, there are many statistical test options in StrandNGS to perform differential expression analysis based on the groups that you have and based on the groups that you want to compare, as well as whether your data is following a normal distribution, that is whether it's parametric or non. There are different parametric and non-parametric tests such as DSeq2, EDGAR, t-test, Lima, Mann-Whitney, ANOVA, etc. In this study, we wanted to compare inoculated and mock samples from 3 different cultivars. So that means 6 possible combinations simultaneously. Hence, we chose to perform two-way ANOVA with a p-value cutoff of 0.05 and a fold change cutoff of 1.0. These cutoffs were selected based on what was mentioned in the paper. As seen from these Venn diagrams in this slide, 579 genes are commonly upregulated and 207 genes are commonly downregulated in all the 3 millet lines. We then performed with the differentially expressed genes, or DE genes, that we got from two-way ANOVA. We ran the gene ontology analysis on both upregulated and downregulated gene list. However, commonly significant Go terms in the 3 millet, so significant Go terms came in both the up and down regulated lists. However, the most commonly significant Go terms in the 3 millet lines came up only in the upregulated gene lists. So in this slide, it shows the significant biological process terms, and the common ones are highlighted in green among the 3 millet lines. The Go terms suggest enhanced signaling metabolic activity, and regulatory mechanisms in the millet varieties. This indicates stronger adaptability to environmental stress, efficient nutrient uptake, and potentially improved symbiotic interactions, making them more resilient and responsive to external stimuli. Similarly, this slide shows the significant Go terms for molecular function, with the highlighted green ones commonly significant in the three millet lines. These significant Go terms indicate an active molecular response, suggesting enhanced signal transduction, triggering pathways for defense, symbiosis, and metabolic regulation. The millet varieties, they likely adjust their nutrient exchange and stress responses, optimizing their interaction with fungi for improved resilience and adaptation. Now, in this paper, the authors looked into the AMS conserved genes from Arabidopsis, and out of the core set of AMS conserved genes, 26 orthologs were also seen as significant between the mock and inoculate groups in the three foxtail millet lines in our study as well. This slide shows the list of these 26 AMS conserved genes. The highest number of genes were seen in the cultivar TT8, that is 25 genes, and the lowest number of genes was seen in IC36, that is 17 genes. Here also 16 genes were commonly significant among all the three millet lines. And among these 16 commonly significant genes, 3 genes highlighted here belong to the ABC transporter family. The ABC or ATP binding cascade transporter family is a large group of membrane proteins that use ADP hydrolysis to transport various molecules across cellular membranes. Hence, we specifically looked into the expression pattern of the 3 conserved genes from the ABC transporter family. And these genes are seen to be upregulated in the inoculated samples compared to the mock samples. And this pattern was quite significantly observed in the cultivar TT8, which indicates that increased phosphorus uptake and distribution by the cultivar due to the symbiotic relationship. In the paper, the authors also looked at the lignin biosynthesis pathway from Arabidopsis, which is a branch of the phenyl propanoid pathway. The lignin biosynthesis pathway specifically diverts intermediates into monolignol production, which is then polymerized to form lignin, which is stored in the cell walls. Now, lignin production in symbiotic plants is like a double-edged sword. It provides structural support and defense while maintaining a favorable environment for beneficial microbes. So proper regulation of the lignin biosynthesis is essential to maximize the symbiotic effects without compromising the plant's health. The lignin biosynthesis pathway for Arabidopsis was available and we downloaded it from KEGG, then imported it into Strand as a gene list. This was then compared with a set of genes in each of the millet lines, a set of upregulated genes in each of the millet lines that you see here. Not upregulated, sorry, fold change fold change genes up or down with each of the, from each of the millet line. And then we saw that 22 genes are common between the pathway and the millet lines, including the CAD enzymes. The CAD enzymes catalyze the final step in monolignol biosynthetic pathway that we were talking about in the previous slide. They convert cinnamaldehydes into cinnamyl alcohols, and these cinnamyl alcohols are precursors of lignin, which is deposited into the cell walls. Here also TT8 exhibits the strongest transcriptional response to fungal inoculation with significant upregulation of CAD genes, suggesting enhanced fungal compatibility. Hanavalval shows the moderate activation, whereas IC36 displays minimal gene expression changes, indicating a weaker response to fungal colonization. Coming to the key findings, we identified about 11.2K differentially expressed genes in foxtail millet, including 26 AMS conserved orthologs, with 16 genes being consistently induced across all three millet lines, suggesting their role in core regulatory mechanisms. Among the cultivars, TT8 exhibited the strongest AMS response, outperforming Hanavalval and IC36, and was enriched in phosphate transporter related processes, offering insights for breeding nutrient efficient and stress resilient millets. After the UN declaration, millet-based analysis has significantly matured in the past year. And in this study, with Strand enabled us to perform a comprehensive transcriptomic analysis in foxtail millet. Our platform can be extended to study other millet varieties as well, providing valuable insights into their genetic potential, adaptive traits, and agricultural resilience. So just like we were able to get inference of TT8 being a better symbiotic variety compared to the other two races, we can find for other millet varieties as well. That's all from my end. Thank you. And we can have the Q&A session now. Over to Khushbu.
Yes, thank you, Vaishali. That was a wonderful presentation. I would request the audience to please raise their hands, or they can even type in the chat box if they have any queries related to the presentation. You're open for the we are open for the Q&A. Yes, Miss Pratima. Hello. Okay, Miss Pratima has raised her hand, but hello. Sunil Cheru has raised the hand. Any question you can please ask.
Yeah, hi. I'm audible?
Yes.
Yeah, hi, Vaishali. This is Sunil
Hi.
here.
Hi, Sunil.
Yeah, so I was curious. So you use that paper as your template to carry out this analysis. Did you find any significant different findings, or much more than what the paper or the authors have reported in their original manuscript?
Right. So, in that finding, we did look at, as I explained in the alignment report part, we did look, it did look like that they might have more differential splicing events or gene fusion events that might be occurring. But we haven't looked at in depth yet, Sunil. And yeah, we'll have to extend that part.
Okay, that's something in pipeline for future.
Yeah, yes.
Okay, thank you.
Thank you, Sunil.
So if no one has any questions, can I ask you one more, Vaishali?
Yes, sure.
Uh, so, uh, s- so the lists In the gene list that you used, you said there were fold change gene lists, right?
Right. Let me just share the screen. It will be easier. The AMS list is what you're talking about?
Yeah. So the when that you showed.
This one?
So yeah, I think so. My site is very bad. But anyway, I was interested. So there is no mention of the upper or lower cutoffs of your fold change.
Oh, yeah. So here the fold change we took were the 2.0.
Twofold.
Yeah,
Twofold.
twofold. This is twofold. But in the differential expression analysis, we did a one-fold analysis because in the paper and also in these, sometimes most of the authors want to find very minute changes as well. So we kept it along like similar to the paper. But in the AMS conserved genes, when we were looking into specifically these genes, the fold change was 2.0.
Okay. So 2 and above or 2 or below. Okay.
Yeah, and you'll be able to see
And
quite a clear, quite a clear higher
yeah, yeah.
extension in the TT8 in
Yeah, yeah.
couple of genes.
So I saw that. So just for my idea, what is the total number of at least annotated genes for this fox millet?
Hmm. I think that Anuksha will be a better person to take that up question. How many genes were there, Anuksha, in that
Yeah, so in
all
the
entities list?
paper they have found over 2000 genes, but according to us, we found a little lower than that number.
No, not the AMS
No,
concept.
no, no.
How many genes are there in the reference or the genes or transcript annotations?
I'm not really sure of the exact number, but
Yeah,
I
that's okay. That's fine. We
But
can find it.
there were approximately 11.2 differentially expressed.
Huh,
So I'm
huh.
assuming there will be at least 20K.
Yeah,
yeah,
that's
I
what the
want.
number is quite
Quite high.
significant kind of DGs.
Yeah.
So I was thinking, what is the starting point?
Yeah, the starting point here was all genes, but again, as I mentioned, we took fold change 1 here
Mhm.
because again, most of the, authors, they want to look at very minute changes, and there were approximately a little bit more differentially expressed genes in the paper, actually.
Okay, and last thing, I'll not hold you for long. So the land races that you mentioned are like native to India or somewhere else, or what's the
So
geography?
I
these
think,
were
yeah.
taken from Taiwan, actually.
And the cultivar?
Both are originated in Taiwan.
Okay, thank you.
Minute Shakti. Yeah, we have few questions in the Q&A session. Can we address this? Yeah. Okay.
So, Dr. Jignisha Patel, what is the significance of this study? Kindly give brief. So significance is what I mentioned in the key findings. We found out, so basically it helped us to understand the symbiotic relationship, how the symbiotic relationship of the fungi is affecting millets as well. And the TT8 demonstrated a strongest AMS response. And we can say that it's the most it was the cultivar was most easily able to take up phosphorus higher phosphorus uptake distribution due to their symbiotic relationship, and that can be further looked at when we are breeding or creating new varieties. Pratima. Okay. Sunita has raised a hand. So Sunita, can you please put the question in chat, Q&A or chat? It will be much easier. Or maybe Khushbu, you can unmute Sunita, whatever works.
Yeah, so we have few questions in the chat box. We can take those up, I believe.
There is one question from Pratima Kochar. I was speaking, but it was not audible to you maybe. My question is, what are the further prospects for future regarding millet research? So for future prospects regarding millet, since we want to, for any crop, we want to find any crops or millets that are more efficient in their stress. They are more tolerant towards stress and they are more efficient in nutrient uptake. So it will be this is like a starting point with from known data sets or known data points. You can find out what kind of what kind of breed or line shows a better response to that stress because of that relationship. And then, of course, further analyze your own data and see how we can get a new, better cultivar or a line better than already that is present. And Srikanti, do you want to add anything on this? Okay, I believe there might be some audio issue.
Yeah, is there a problem?
Khushbu, there is a hand raised by Sunita Chauhan. So
Oh, okay.
would
One
you be
minute.
able to
Yeah,
unmute?
yeah. Yes, Miss Sunita, you can please unmute yourself.
Yeah, Namaskar, ma'am. I have a question. Like you have told about the symbiotic relationship with the fungi and the millets. Like suppose I want to do some industrial application studies with millets. So suppose I put some fungal treatment on the millets and want to check out the like what are the changes in the proximate analysis parameters, like what is the percentage of cellulose now and lignin content and this kind of study can you done?
Yes, we can, but I would like to request Srikanti to pitch in on this.
I think Srikanti is unavailable. I can take it.
Yeah.
Charlie.
Please, yeah.
Yes.
Thank you.
Sunita, right? So you are saying that you
Yeah.
have different varieties and you want to study those plants. Is that what is your question?
No, ma'am, I want to study the effect of different fungal treatments on this particular plant waste.
Okay, okay.
So suppose I treat the waste or the plant material with that particular fungus, like 3-4 types of different fungal treatment of the millet plants. Then I want to analyze what is the effect of fungal treatment on the plant, same variety of millet, na? Using
Right, right.
different fungi. Like, can we do through dry end application? Like, can we analyze how much lignin is changed, lignin content, cellulose, hemicellulose content through your dry end study? Like, generally, what we do proximate analysis in our lab, wet end study. So instead of going through the wet end study, can we go like that?
So here they have given a proof of concept principle by taking. So as Vaishali mentioned, we couldn't share the ones where we have done experiments because that's confidential, right? Because it's client study. So if you have enough publications which is similar to what you want to study, and if the data is available, then we can do a dry analysis or bioinformatics analysis to explore options and to find any novel findings from those studies. But if such
But
studies are not available, sorry.
you need samples not treated. those treated samples to extract the DNA, RNA, जो भी आप करेंगे, whatever.
Yes. So we do end-to-end analysis. So if you have performed experiments, we can take up those
Yeah.
samples, we can analyze and give end-to-end data from.
Ma'am, what is the quantity required for you?
So if you're saying plant sample and if you're sending us DNA sample, so for any our DNA analysis for genome sequencing, whole genome sequencing, we would need 100 nanograms. So in this particular study, they have done RNA. And if you want to study RNA, then you have to send RNA around. It depends on whether you want to do only look at mRNA or you want to look at non-coding RNA as well. Depending on that, the protocol will vary and we would need different amounts of RNA for each of them. So
Okay.
the
So,
minimum amount, sorry. Yeah, please go ahead.
We will not be able to give you the RNA or DNA. We will give you the treated samples.
Yeah, we can take up that as well. We can do extraction at our end and then we can
So that
provide
is
you.
what I am asking कि how much quantity you require treated samples की वो तो nanogram में नहीं होगा. You want half kg or 1 kg.
No. So we need milligrams amounts. So anywhere between 50 to 100 milligram samples or
Okay.
if it is, yes. Yeah.
Oh, this much only. Okay. That's great. That's great. Ma'am, I would like to go in more details.
Yeah,
I will write.
you can email us and we
And
will be able to get in touch
what
with you.
are the charges profile, like how, and what is the time required for this kind of analysis at your end? So I would like to see, because generally we are doing it at our wet end now. So instead of going through, if we are saving time and much energy, like you are saying, it requires only milligram of sample, then it's very good. So we can go ahead with that while optimization studies. Now, instead of going through the wet end study, we can go like that. So I would like to have the, like,
We can
what will be the
connect
charges?
offline, Sunita. So we can connect offline and our sales executives will reach out to you and we can
Yes, ma'am.
connect and we
Sure.
can discuss this in detail. So we can see how we
Oh,
can help.
Ji. Thank you. Thank you so much.
We can take up the other questions, I believe.
I can't see any other question in the Q&A, Khushbu. Yeah, Dr. Patel, she's mentioned, could you please share the recorded video? So the video will be uploaded in YouTube and it will also later be a part of the website. So yeah, we can share it with you.
Sure, Vaishali, I will share it. Yeah, I'll share it.
Okay,
across.
ma'am. Could we leave? Thank you. It's over now.
Are there any more questions from the audience? Like, yeah, we have resolved the ones that are there in the chat box, but then does anyone want to like ask? Okay, I believe that's it there. Okay, thank you everyone. Thank you for joining. I guess there's one more person. So sorry, Miss Sanchita.
Yes, actually, I already have a project with you all, and
Okay.
Haribo, Srikanti, they all know me. So I just, because in the beginning, it's not related to today's presentation, but in the beginning, as you said that, product development was one of the areas in which you all work as well. So I was just wondering, like, what kind of product do you develop? Like you act as industrial partners for projects, or like what did you mean by that?
I meant about the products that can be, in dry lab as well, like software or any product specifically based on your requirements. It can be also a kind of a limb successioning dashboard. It can be a software product, that kind of product. But is
Okay,
there
and
anything specific that you're looking for?
no, I think this dry lab product is fine, but like, is it like some kind of AI related algorithm or something? Do you all do something of that sort as well?
So we have a couple of projects that are going on in Strand for which are based on AI and ML. So if there is any interest to you, we can send you some details as well on that.
Yeah, not right now, but I was just trying to understand about the product development part. So yeah,
Right. So
thank you.
I mean, it's based on the user requirements, or even like internally, if there is any research requirement, we have that. We have like almost 6-7 MLAI projects that are happening, and there's one that is being presented in Bio IT World in US as well soon, next week. So if
Yeah,
you
okay,
have it,
thanks.
we can send you the details.
Yeah, sure. I'll get in touch with your team when if it happens, if it ever happens. Okay.
Okay,
Thank you.
sure, Sanjita. Thank you.
Yeah. So thank you, Ms. Sanchita, for the question. And thank you, Vaishali, for addressing all the questions so nicely. And thank you, Anoksha, Pooja, and Shikanti as well. She was also there in the webinar. And also, I would like to thank Arivu for joining. Yes, thank you everyone for that wonderful time and engagement. And yes, Vaishali, we hope we do more webinars ahead with you and your team. Please come up with such researches and engage us more. Thank you.
Sure. Thanks, Kushbu.
Yeah.
Thank you for conducting it nicely.
Yes.
Thanks a
Thank you,
lot.
everyone.
Thank you, everyone.